video production on the run. This is fun. Science experiments is what we do here, and this is an experiment as well. Even though it's our second time here in terms of doing a live stream on Giving Tuesday, so welcome back. This is Giving Tuesday live at the Boyce Thompson Institute, and we are here celebrating all of the wonderful science that we do here at BTI, and maybe even putting our hand out a little bit in support of science, like we're going to hear about right now from members of our FAY Lab. And so we have 5 members of the FAY Lab. We had to expand our set a little bit here, but we wanted to bring as much of the lab perspective as we could here. And you might be familiar with some of their work. There's made some big headlines recently regarding apple and pumpkin research. So let's start off and try to project as much as possible as we continue to work through our audio. Let's just go around the semicircle and just give a brief introduction of who you are and maybe how you got to BTI and what you do in the FAY Lab.
Okay, so I'll start first. So I'm Xiaopeng, so I'm a postdoc in FAY Lab, and so it's my second year here. So I got my PhD in plant pathology. So my PhD work was associated with plant biology of plant pathogens using genomic and transcript data to study the biology of fungi. So after I got my PhD, I realized that I need some extra training in plant science because most PhD work was associated with fungi. I actually have no idea about how plants function work.
Yeah,
that's
a big part of it.
That's why I come to BTI and do my postdoc research. So in phase batch, because bioinformatics is my interesting aspect. So I got a lot of training in bioinformatics during my PhD student, and also 2 years visiting student in Cornell in another lab. So yeah, that's the reason I come here. So about the project I'm working on is I have a couple of projects working on, but one project I think very interesting is to discover viruses in plants using small RNA sequencing. So we actually have 2 people working on this project, and he is also a postdoc in lab, but he's not with us today. But I can introduce this topic and some of focus done by E. So the purpose for this project is to identify some viruses in plant using small sequence data, because plant can respond to virus infection by triggering some defense mechanisms, which can generate some small RNA, which so-called virus-derived small RNA. So this small RNA can be captured by next generation libraries, and then we can do sequencing and de novo assembly and classification. So that's our main job. So the strength of this method to identify viruses is resolution. It can discover many hidden viruses that's probably in low titration in plant cells, but often missed by traditional method. So and also, it can also discover some novel viruses. For traditional method, if you don't have some prior knowledge, it's very difficult to find these novel viruses. So this method can provide this opportunity to get this virus out. So our job was first to develop a pipeline, because for many people in our lab, the sample collection and library preparation is kind of easy, but they probably have some problems with data analyzing, especially if they have huge samples, like our sweet potato samples. We have 1,000 samples collected from everywhere, so it's difficult to handle such a huge data.
It should be known also that everyone here works on computers.
Yeah.
So we're not your traditional scientists. None of us are in wet lab. we come wearing collared shirts and sit at a desk. It's pretty good. It's a pretty good way to do science.
Well, after we do introductions, I'm going to have someone give us the elevator pitch about what is bioinformatics.
Oh, wonderful.
So your role. Right.
Yeah.
Yeah. Don't tell too much. Don't tell too much.
We have to do. We need a dramatic reveal here.
Oh,
is what we're, no, I'm just kidding.
Behind the, I guess the lab door. Yeah, there we go.
Yeah, something
behind
like
behind
that.
the lab door. Fantastic.
Yeah. So we also we develop a pipeline so people can just upload their raw data and get various result back in hours. That really depends on the scale of data. So for people who is not familiar with Linux environment, we also developed an online service. People just upload data via web page and get result through several clicks. So it's very simple. So with this pipeline in hand, we also do some basic discoveries in our own project. We have a sweet potato viral project. We collaborate with other people who collected 1,000 samples. So and also for tomato samples. So yeah, in both projects we successfully developed several new viruses, and yeah, that's something about our viral project. So I'll hand it over to you.
Sure.
All right, great.
Yeah.
Thank you.
Yeah. I'm John Mendieta. I started here. John Pablo Mendieta. I started here 6 months ago, which went by incredibly quickly. Yeah. Before that, I was working at CU Boulder, doing similar stuff, doing bioinformatics, looking at plants. Yeah, and then I applied to a job here, and it's been great ever since. Yeah, my skills are, I guess, I was trained traditionally as just a wet lab person. Hated it, terrible at it. Pipettes are my nightmare. And yeah, started taught myself computer science, and now I'm here.
So if it's also your nightmare out there, there is hope.
There is hope.
There's hope after the pipette.
There is. It's a much more fun realm of biology, I think, especially if you like computers, because yeah, who doesn't at this point?
They're important.
They're useful.
They're catching on.
Yeah, they're really gaining traction.
Yeah.
What I've been working on in the lab is I've been looking at genetic variants in apple genomes. So everyone likes apples, and there's a huge amount of variety and diversity in them. So what we've been doing is just looking at all the differences in all these different cultivars and trying to understand what do these mean for the apple genome. What makes some apples sweeter or some more sour, or what gives them different pigmentation colors. And yeah, and apple's a really cool crop. You can actually go back and eat varieties from the 17th century. You can go back and see impressionist paintings created by Van Gogh and some others about these apple cultivars that you can go out and pick. That's pretty freaking cool, if you ask me. So that's kind of one of the big projects I've been working on recently. So
Great.
yeah.
Now it's me.
Now it's you.
It's all you.
Okay. I'm Chen Jiao. Now I'm a postdoc in the lab, and I have been here for more than 4 years.
Okay.
So I'm the
Senior.
Yes.
Are you the boss?
No.
Very soon.
So the first 2 years I'm here, I was visiting student.
Okay.
And originally I came from a wet lab from China. So at that time I studied gray powder mildew resistance. But then I came here, I started how to do bioinformatics. Then I started to love this
Field.
This field.
Yeah.
This science.
Yes,
This
I started
science.
to love this field.
Yeah.
So now I'm working on several, I think, important projects, some genome project and some epigenome project and some transcriptome data. So now I want to introduce an important and interesting project that's genome and spinach genome and the transcriptome data project. So spinach is really a popular green leafy vegetable today.
Yeah,
so
it's good.
Yeah, it is.
It's in all my smoothies.
Yes.
Something I've come to love as an adult. Not so much when I was at high, but
Underrated.
Yes.
It gets a bad rap.
it does.
Now people pay attention to their health, so
Yeah.
they know that it is a rich source of various nutrients like carotenoid, folate, iron, and calcium. So
You sound a
it
lot
is-
like my mom right now. just so
I'm
you're
always
aware.
like
Eat your
your mom.
spinach, John. Eat your spinach.
Yeah. Come on, man.
Look what it does for Popeye.
Yeah. We put all this money into the genome.
Is there a better
Eat it.
spokesperson for spinach than Popeye? Is he still the go-to spinach? Like when you look for someone that has made spinach famous,
I think
I feel like it's got to be
I
Popeye.
feel like we need to update that like deeply. Like the last spokesperson was in like 1940s,
Yeah.
so there's a gap right there.
Yeah.
That's maybe,
Jolly Green Giant, does he
That's
do
what I
some
was gonna say,
spinach?
yeah.
That's a good
He
one.
does some spinach. Okay,
That's a
so
good
we got.
one. It's just
Okay.
it's still
We
out.
could use a refresher though.
Yeah, yeah. So
But
new,
anyway.
new branding, new marketing.
New movie or something.
Yeah. So
I'll work on that part. You guys
Okay.
just keep doing the research.
There we
I'll
go.
look for the mascot.
There we go.
So it is beneficial food for our body, but itself is facing several serious problems like some abiotic stress and biotic stress. Abiotic stress, like very high temperature, it cannot tolerate that temperature, then it will die. And some serious pathogens like Dunning-Mildew pathogens. So and the resistance to Dunning-Mildew pathogens is always the breeding focus for the entire spinach community. But now we want to provide more available genetic and genomic resources for the community to facilitate their breeding progress. So we sequenced the genome of spinach cultivar and transcriptome of 120 transcriptome of cultivated and wild spinach. So we finally assembled a good quality genome and provide more than 200 variants across all the analysis the spinach. So using those findings, we can study the spinach evolution, domestication history, and also we identify the selective sweeps associated with various important agronomic traits like bolting, flowering time. So the data is released this year, and since then we received a lot of emails asking about our data and about our materials. And they are not all spinach group. They have their own study focus, and I'm glad that we can help them to of their research. So now we are working on improving the genome quality of the spinach. So now we get some extra third generation sequencing data, and the result is good. And I think I and Pablo will continue working on improve the genome quality to further facilitate their breeding progress. Yes.
Great.
Yeah.
There you have it, Fay Lab and spinach. We'll try to, we're going to play, I'm going to test myself in front of all of you to see if I can name all the crops in which the Fay Lab has touched.
Dude, good
in
luck.
recent times.
I work here.
I'm
I
going to
don't
try.
know.
I'm going to try because my failure will be education for the audience. So
Yeah.
that's good TV. I don't know. All right.
So
Next.
it's my turn.
Yes.
Okay, my name is Shan Wu. I'm a postdoc in Fay's lab. I received my PhD from the Ohio State University.
The
yeah,
Ohio
they
State,
have
right?
the the.
Yes, you have to say the.
Yeah,
you're
the
told.
Ohio
That's
State.
like a-
Oh yeah, oh yeah.
I didn't know that.
Yeah,
Yes.
the Department of Horticulture and Crop Science. And in 2015, and same year I joined Fay's lab. During my PhD, I was working on a project collaborating with Dr. Fay and Dr. Catala's group in BTI on studying tomato fruit quality trait. So I was trained, my training was focused on molecular biology and plant genetics. And during my second half of my PhD, we started doing a lot of RNA-seq, which when the RNA sequencing become more affordable and then I started to be interested in learning bioinformatics by myself. But I really was a bench scientist before I came to Facebook. And now I have all the experts who can help me.
Yeah, now you're on computer.
Yeah.
Slowly transitioning people.
Chen helped me a lot. Whenever I have questions, I just bother her. And, yeah, I bother everyone. Yeah.
Hey, that's how you learn.
Yeah.
I mean, I'm bothering all of you right now. You should be doing work, but I'm bothering you. It's for a good cause.
It's
Almost lunchtime anyway.
Well, we're going to drag it out to lunch.
yeah. It's a really good learning and working environment in Face Lab. And so I'm currently working on several de novo genome assembly projects. This year, we actually have 2 publications out on 2 crops in the Cucurbitaceae family. One is pumpkin, the other one is bottle gourd. So
Very seasonal.
yeah,
Very seasonal.
yeah. So yeah, it's right before
Halloween.
yeah, the Halloween.
Yeah.
So yeah, so maybe I should talk a little bit more about the pumpkin genome.
Sure, let's talk about the pumpkin genome.
So yeah, so the first goal of the pumpkin genome project was to provide a high quality reference genome for pumpkins. Pumpkin is an important crop. You can eat them, but also it is an important rootstock for other cucurbit species. So like you can graft watermelon and cucumber onto pumpkins because they're more disease resistant.
I didn't know that.
Yeah.
I'm also learning in this
Yeah,
lab
I mean,
all the time.
Imagine if you shared an office or something.
Imagine if I sat next to these people. I'd absorb so much.
Yeah.
Well, is that a good like at-home gardening tip? Putting
Can you do that?
pumpkin material, or is that
If
does that not
you're-
really translate? Does that work?
Yeah, I don't
If you
know.
grow in, do you grow? I don't know if people grow watermelon and cucumber at home, or at a big scale, or in a soil that not super appropriate for growing those
Oh, okay.
crops. Then it's going to be good to use rootstock
Okay.
that more resistant to biotic and abiotic stress like pumpkin.
Cool.
So, and also bottle gourd. Bottle gourd is also used as a rootstock for other cucurbit.
Well, I feel like bottle gourd's a weird one too, because you don't see that much in like North America. Like I came to the lab and I didn't really realize what bottle gourd was. Yeah, and it's just like, so if you ever see old drinking gourds in like, I don't know, cartoons or something, that's what it
Yeah.
is. It
Yeah,
looks
but
like a bottle.
Yeah,
That's-
it is an important rootstock because it's resistant to a lot of virus and also abiotic stress.
What does it taste like? Have you eaten it? Can you eat it? You can eat it, right?
Yeah, I guess. Yeah.
It's bitter? It's pretty...
It's not bitter, I don't think.
I think we use that as a container in China to
Yeah, that's,
hold the water.
And,
Oh, really?
And also
Yeah.
decoration. For
Yeah.
The, yeah.
You can buy them on Etsy. Like, Etsy, get at me. Yeah, you actually can.
Sorry,
I
you
was
missed
curious.
Cyber Monday.
Damn it.
So providing a high-quality reference genome is very important for breeding of these important crops. Yeah, so and another thing about the reference genome, like high-quality genome assembly, is that you can do comparative analysis and then study the evolution history of these plant species. So from our pumpkin study, we actually, by comparing the pumpkin genome to other cucurbit genomes, we found there's a allopolyploidization event, specific in the pumpkin lineage, but not in the other cucurbit crops like.
Would you like to
What?
explain what an allopolyploidization
Oh,
event
okay.
is? Because like I didn't know until I showed up here.
I was going to ask it, but I wouldn't be able to re-pronounce it.
Yeah, I feel
Right.
So,
It's,
I totally
thanks for
feel
stepping
that.
in.
Yeah.
So it's like 2 diploid species came together, become a tetraploid. And of course, after years and years of evolution, it diploidized to a diploid. So we consider the modern pumpkin as a diploid. But like maybe it can go back to it's between 26 million to 3 million years ago. That's when the hybridization happened. And then it's 2 different species joined into one. That's called allotetraploidization. So,
It's a long history.
yes.
For the
But
pumpkin.
by analyzing these high-quality genome, you can identify these events happened way back in history, and then study why this is beneficial to the species, why the species is successful and survived, maybe after some disaster. Only
Yeah.
this tetraploid survived, so we can learn from that, and then it can help us better improve our crop today.
So in a lot of ways, you are all sort of like genetic archaeologists.
Honestly, that's like a pretty good way of putting
Yes.
it. Yeah,
Yes.
no.
That's pretty cool, right?
Yeah,
Is that-
it's like
Yes,
one of
we
the.
can go back to history and study the genome and see what happened.
yeah.
Totally.
Yeah,
That's
really
fun.
cool.
Yeah,
You
it's-
need, you need like your Indiana Jones equivalent.
Yeah. Well, I've always thought that like agriculture was like a perfect it's synonymous with civilization, right? As our ancestors domesticated grains and barley, we were domesticating ourselves from hunter-gatherer lifestyles. So it's like it's two sides of the same coin. The plants domesticated us, and we domesticated them. So it's agriculture is cool. It's deeply human in every way. I look at it.
Yeah.
Yeah.
And another project I was involved in is sweet potato. Maybe that's connected to Ryland's project very well. So we are also writing the manuscript right now. We assembled two diploid progenitor genomes of the hexaploid sweet potato. So the sweet potato we eat right now is a hexaploid crop. The genome is huge and it's very complex. So it's very hard to assemble it, de novo assemble the hexaploid genome. So our approach was to assemble the diploid species that's closely related to sweet potato. And then, yeah, we're working on that, and we'll have a story out very soon.
Okay.
BKG, don't tell them anything. Tell them nothing.
Cliffhanger.
Tell them
Cliffhanger.
nothing.
Still an embargo.
Yeah, right?
I feel like this is
And
awkward.
finally-
Yeah, I feel like this is awkward because your back's almost like towards the camera now. So maybe
No, it's okay.
Later.
No one needs to see me.
Yeah,
They're not here for
I,
me.
I have that same feeling every day of my life. It's okay, and here I am in front of the camera.
Nice, dude.
A lot of caffeine and halfway to spastic, and
Yeah, no, that's good. I mean,
Dude, that's how you do science.
Yeah.
It's
Yeah.
like that coffee pot is everything.
oh, the coffee pots. I mean, gotta love BTI. Shouts out to the second floor coffee
Coffee
bar,
machine.
right?
Yeah.
I've actually heard that a number of BTI collaborations, the catalyst for those internally has been the coffee pot.
Seriously?
People get talking, different labs start talking at the coffee pot
And-
about their research, and it's like, hey, I might be able to help with that. And
That's dope.
I think Frank Schroeder and Dan Klessig had a story like that. Now they're kind of collaborating on something because they just happen to get talking over the coffee pot. So
That's insane.
all good things happen over coffee, and we also do coffee research here at BTI.
We do.
Yeah.
Plug. Yeah.
BTI, btiscience.org/give.
There we go. Nice.
Awesome. If you like coffee. Just saying.
Well,
Let's go back over here.
Fine. I'm Ryland.
Hi, Ryland.
I'm a grad student in Fay Lab. I joined over the summer, so I've been here a couple weeks. What's the opposite of longer? Shorter. That doesn't sound right. Than Pablo has.
Sure, like the same time.
Joined right around the same time. Yeah. So, and this is my first foray into bioinformatics. I was purely molecular up until 6 months ago. So, pretty cool. It was challenging at first, but then again, what isn't challenging in science?
Right.
So, I was again terrible at molecular biology. I don't have a lot of patience, kind of ADD. So bioinformatics was really conducive to. It's very evident if something doesn't work, and if something does work, you get instantaneous results or instantaneous feedback at least, and you can just. It's very high throughput, so you can keep going, and it's conducive to that lifestyle. So it's pretty cool.
Yeah, we run experiments like daily,
Yeah.
which is super nice. It's not
Yeah.
like we don't have to wait 2 months. It's like I run multiple experiments every day.
Yeah.
Experiments, whatever.
We're all involved in like too many projects to talk about necessarily all in one sitting, so that's cool. I mean, when we were talking about how many organisms does the Fay Lab looked at, how many species, it's probably too many to count, probably too many for you to list. So.
Probably.
It's gonna be a good game.
Still might try.
So I guess when I'm not in class, I am in Mulham, and or at desk. And I'm currently involved in doing research on sweet potato gene expression and how it is influenced by viral infection. So looking at several different viral treatments, and in conjunction with one another. So separate viral treatments in conjunction with one another, how they change expression. So looking at kind of biotic stresses, and currently just looking at changes in gene expression related to mRNA, but we also have small RNA data as well that we're going to be looking at as well. So doing a lot of that. I mean, I'm in the plant pathology department here, so it's great to be able to join a lab that has such a heavy.
Fei Wei Li, you'll see him at 5 o'clock.
5 o'clock, hashtag Fern
Right
God
here.
in the building. Fern God.
Fernway on Twitter.
Is that really?
Great
That's right.
Twitter handle.
That's a great Twitter handle.
Yeah.
Yeah, so I mean, it's great to be involved in a lab that has not only can be able to assemble these genomes and has such just a hand in every pod. So I get to do pretty much exclusively plant pathology in a lab that does everything. So it's pretty cool. Yeah, I don't know. I mean, everybody went around and talked about their projects. I don't need to necessarily talk too much more, but it was, yeah, it was. I don't know.
Okay, well, you've all explained a lot about what you do, so I'm going to try to, if I'm someone watching and maybe I don't understand everything you're talking about, I think the first question I might have is, so you all talked about you work on computers, right? So so when they say wet lab, you're not in the actual lab working with chemicals and solutions. You're looking at data and you're working on computers, and that's bioinformatics. So who wants to define bioinformatics for us? What is that? What does that term really mean?
Maybe you can start.
Sure. Yeah, I mean, I kind of see bioinformatics as it's biological information, right? It's everything that you can extract from the DNA or the way something looks and processing that using computers. So anything dealing with biology and computers is at that interface that we're dubbing bioinformatics. It's also, so it's using a lot of computer science to actually unravel how biology works and how biological things function, because 2001, we sequenced the human genome. We thought that was going to be it. We thought we'd understand everything. And we're in 2017, and we're still trying to figure out most things. We know a lot, but there's a lot more we don't know. And computers are a great way to figure that out, because they're fast, they don't require months, and they allow us to get at really large-scale questions instantaneously. So, yeah.
And part of the need for bioinformatics, correct me if I'm wrong, is there has been such an acceleration in various technologies that you're now getting so much more data back. And now scientists who made, wet lab scientists need people like yourselves to take those huge amounts of data and actually, decipher, translate, whatever you want to call it, and make it usable.
Yeah, I mean, I feel like there's just a massive bottleneck at one point where it's just there's all this phenotypic data and genotypic data that's kind of just circulating in the community. Not a lot of people that are able to process it, and so, I mean, it's really slow moving until you can start applying computers to it, and then.
Well, it's also crazy because it's like the people like you two are like some of the first generation of bioinformatics scientists that we've actually seen. Yeah. Right? Like you guys were.
Did you know that? You look surprised.
I mean, it's true.
Yeah, like we came from the wet lab, and I love wet lab work, and I'm good at it.
We're not saying anything bad about the wet lab.
Gifted.
We're not saying anything bad about it.
Yes, and I like
Steady
plant
hands.
genetics.
Yeah.
But for some question, you have to use the bioinformatics to solve your problem. And then I think it will eventually become a standard, routine for biology.
Yes.
Yeah.
Just like PCR in the lab.
Yes.
And you have to have a people working on bioinformatics, even not in dry lab, but also in wet lab. And you can split your group into two or many subgroups and
Yeah.
people working on
Two very
children's,
tribal
yeah.
pockets, the computer people in the wet lab.
Yeah, I think bioinformatics is a field involving many subjects. So basically, it's biology and informatics. So you are looking for some signals from biological data. So then how to explain this data, how to deal with it. Because in old years, people mostly working on handful data from amino acid, protein, and nucleotide. But now we have more data and huge data generated every day. So how to analyze data, how to store it is still a question. And yeah.
Well, it's weird, right? Like the idea of being able to download hundreds of genomes on a computer.
Yeah.
20 years ago, that was a phrase, that's science fiction.
Yeah.
We're in 2017, and that's now. You can just go do that.
The cars aren't flying, but we can do that.
Yeah, we can do that, which is like...
It's a good give and take.
Yeah, I think, I like it. I think the genetics is cooler than flying cars.
Yeah.
But I haven't flown
That
in a
would just
car.
be a headache anyway.
I think so. Afraid of heights.
Yeah.
I'm not. Sorry. But it's a pretty amazing field to be in because it moves faster than anything. It's like, I read papers from 2014 and they feel old. And I don't think there are many fields of science that feel like that.
Yeah.
So it's...
Well,
It's
that brings
right.
me to my next question, and this will be kind of the last basic question, but I think it's important to help not only define what you're all doing, but help gain an appreciation for it as well. So when you say, I sequenced this particular genome, what does that mean, and why is that really important to the industry?
Yeah.
So, yeah.
I'm looking at you. Looking at you.
Yes, when you have the genome, for instance, our bottle gourd project, when you have a high-quality genome, you'll be able to identify genes that associate with an important trait, for instance, virus resistance. And you'll be able to map it and develop, map the genome. Then you can study the function of a gene. That's for the scientific aspect. But for the industry, you'll be able to develop a marker that can link to that trait. So you can do selection, marker-assisted selection, more efficiently to move the population forward and to develop a better variety. So I think that's the direct use of a high-quality genome assisted breeding program.
Higher yield, more nutrition,
Yeah. And
viral resistance.
It's a bit of like kind of
pathogen resistance.
resistance.
Yeah.
yeah, you can breed, do your breeding more efficiently, much more efficiently.
So once you've mapped or sequenced a genome, would a good analogy be to say you've kind of pushed the first domino over, and that can make things happen a little bit faster?
It's certainly the
from
start
there?
of a long process. It's the first
Maybe
step.
not rapidly like a domino's falling, but you know what I mean? It's kind of like it maybe opens the door to a lot more.
Yeah.
Dominoes falling in zero gravity or something like that.
Sure.
All right.
Yeah.
I like it. I like it. It's the first step of allowing a crop to be modernized. And what that really means is it gives breeders the capacity of selecting for really specific things that are really important to a lot of people. Genetics is hella complicated. I don't think any of us know all of it.
Come on.
Okay, I know. I know
Don't be
all of
modest.
it. Yeah, I know literally everything. No, but it's
There's a Nobel Prize in this group eventually, I think. Right?
Dude, don't look at me. Don't look at me.
No
That's
pressure.
about luck.
Yeah, I think it's a little. All great. Yeah, all truly miraculous findings are pretty lucky, I think.
Yeah.
Or maybe that's just bitter scientists saying that. I'm not sure which.
Well.
But it's certainly the beginning. Like a well-sequenced genome is certainly the beginning of like deep dives into evolution, as well as the first step towards modernization of a crop and improving it for the modern age. Climate change is scary. Population expansion are scary. But with these things, we can start working towards combating those things.
Yeah, and that really gets at the environmental and food production side of BTI's mission.
Totally.
Is I mean, these are real problems. And the solutions start at your keyboards
Yeah.
frequently.
Yeah, I mean, curing cancer doesn't matter if you don't have anything to eat, right? You need to be old to have cancer.
I mean, we can try to do both, right?
Yeah.
I mean,
Yeah, we should probably do both. We should probably do both. But yeah, food's essential. And it's cool to work in a lab that's so multifaceted.
Okay, so speaking of food, here we go.
All right.
I'm going to try to name all the crops I know that Fay Lab has tackled. At least, I mean, again, I've only been here a year and a half, but so I've tried to do some research. So you've mentioned sweet potato.
Sure.
We've mentioned watermelon.
Okay, this is some good gimmies.
Tomato.
Don't tell him anything. Let him struggle.
I feel like everybody at BTI has tackled tomato. In different degrees.
I feel
it's what they call a good model organism. Is that fair?
Jesus. Nice.
Cucumber. Is that
That's
right?
Yeah.
You mentioned gourd and pumpkin.
Yep.
We mentioned apple,
Yes.
spinach.
Yes.
Watermelon.
Don't let him struggle.
Okay.
Grape.
Yeah, sometimes.
Okay.
We collaborate.
I didn't even know that.
Okay.
Senior one.
Yeah.
Boy, it's starting to get tough now. You talk. I look less knowledgeable because you all discussed all those crops already. Watermelon, grape.
Do you know anymore?
What did I miss?
It's close to melon. Watermelon. It's melon. Yeah,
So there's
like a
watermelon and melon, right? So
Oh,
there's-
watermelon and melon.
Yeah.
Okay.
Different things apparently.
Right.
Are we just doing crops?
Just
We
name
did,
things
like,
that-
white fly.
Yeah, we also
White
have
fly,
shrimp.
yes.
Yeah.
White fly, that's a big one.
There's-
That's a big one.
Kiwi is one that the Fay Lab's done.
Yeah.
Kiwi, okay.
Yeah.
I did know that one.
Yeah.
I forgot that one. Yeah.
Pomegranate.
Oh, pomegranate.
Pomegranate.
Pomegranate,
Yeah,
okay.
that's a new one. Jesus. I don't
So
know.
we're doing a lot here. That's the point of my quizzing myself.
Can you
Is,
juggle all of those at once? If I
probably
gave you
not.
each one of them.
I could juggle two.
I do like
That's the
the
extent of my
idea.
juggling.
With one hand or?
No, two hands.
We
Which
should have
isn't juggling.
That's not juggling.
Right.
Yeah.
We should have a lab dinner where we only eat the cultivars we research, because it's still just going to be like a normal dinner. It's
Yeah.
like, oh, look, it's everything.
So a lot of people just had Thanksgiving, so I wanted to bring up all those crops because there's a good amount of those crops are probably on the table, right?
Yeah, totally.
I mean, so when you have a big feast like that at your homes, is it like I'm eating my work? Is that weird? Is it great? Is it exciting?
Yeah, I
Do you
will-
not think about it?
Yeah,
Do
I
you-
would say this pumpkin is mashata. This is
Yeah.
maximum.
My parents kind of hate it. They thought it was fun, and then they're like, just stop. It's like, let's talk about something else. And it's like, no, Mom, this is Thanksgiving. Let's talk about pumpkin.
Can we just eat and talk about football?
Yeah.
You want to talk about the genetics of our food?
Want to talk about the genetics of turkeys?
You're
Come
scary.
on. Yeah, it certainly flavors your world in a different way when you
Okay.
research this stuff.
Well, my final question is just what's next in bioinformatics? So this is a rapidly accelerating
Yeah, field.
industry or field or study. So what are your guesses? Anybody have a guess for kind of what's the next big thing in bioinformatics?
Flying cars.
Look at you, PhDs.
Flying cars?
Yeah, looking at you, PhDs. Come on.
So I think bioinformatics is not just providing tools or pipelines to scientists, but also it itself is a science. And people can bring other subjects to this field and make this bioinformatics more strong. Like people can now follow some more ideas in mathematics and statistics, and then provide like our bioinformaticians more options to establish all methods and tools for our own study. So I think the future direction is to get more attractions from cross subjects and then solve for classical biologists.
Like using machine learning.
Yeah, just like machine learning and other algorithms. So the future direction is make it more complicated.
Yeah, yeah, no,
Make it more complicated. Okay.
it's gonna get harder,
that's
which
good.
is hard to think about.
But it's fun that, I mean, you said it, you're all somewhat on the ground floor
Yeah.
of this thing, so, you know.
Constantly learning.
It won't take long before you are the global experts, you know.
That's the dream.
Yeah. You'll
yeah.
be doing talk shows much better than this.
Who knows? Who knows? I think a lot of the future also is just, I think I see bioinformatics right now as like a toddler. Like as a field, it shows a lot of potential and it's really smart for a kid. And it's learning to walk and it's really starting to support itself. But what I think we're going to see in the next decades or two is it's just going to start sprinting. Because there's a lot of things we do know and a lot of things we don't know. We don't know how many genes are in the human body, which is crazy. We have ideas, we have really good estimates, but we're not 100% sure. And we know what 1% of those genes does. It is miraculous how much we know and miraculous how little we know. And I just see the future as better statistics, better data, smarter kids. Like, god damn, people smarter than me doing this. And I can't even speculate. The next decade of biology is going to blow your mind.
Yeah, you know, Paul Debbie was on before you all, and one thing that he and I like to talk about is this how we visualize research. It's kind of the you're kind of these modern day explorers. You know, that we know a lot about what land masses exist on the globe now, but now we're exploring at this genetic level where it's still very much unmapped.
Yeah.
And so to kind of think of it as, you know, we talked about being archaeologists, but also explorers, right? You're you know, going through these spaces that not very much is known about, and you're trying to see what you can find. You know, you just said 1% of the genes, we know what they do, right?
Yeah.
You know, so we think we know all of them, but we don't really know what they do. And so,
Yeah.
yeah, so there's a lot of work to be done, and I'm excited to see what you all do with it.
Yeah.
It's pretty incredible what you've done so far and what I've learned about just in my time being here. And hopefully everyone watching now has a newfound education of bioinformatics. We'll be talking bioinformatics a little bit later, talking about the new center we're starting up here at BTI. And the last thing I just want to say is a lot of what you produce is open source, correct?
Yeah.
Yeah.
So this is, you know, the discoveries you're making, you're not just keeping to yourself. So if I wanted to harness some of this open source data, is there a place I go to do that? What would be the best way to go about it?
Yeah.
So for some tools we develop in our lab, you can just go to PhilLab website page.
Okay.
And you can search on these tools. And for other tools, we probably deposit in GitHub or but anyway, any tools can be found in the paper. Once we publish paper, we will specify where the softwares are located.
Okay.
So
Sure.
yeah, that's
Qubit genomic database,
Yeah.
if you just want to have some genomes floating around your computer, that's like always a good thing.
All the pumpkin,
Yeah.
cucumber, watermelon, melon genomes can be downloaded from the Qubit genomics database.
pretty
Which
sweet.
is hosted in our lab.
Yeah.
Okay. So there's a number of databases you can access. You can start by going to the PhilLab portion of btiscience.org. Have some fun exploring there. And thank you all for coming on and talking about your work. This is incredible stuff. It's groundbreaking. It's exciting. I think it's really becoming kind of, you know, one of the defining characteristics of BTI.
Yeah, hot young thing.
Yeah.
Yeah,
It's
nice.
all we want to be, right?
That's all we
That's all anyone can aspire for. Well, we thank the members of the PhilLab. Look up a lot of their work. We've done some recent videos and publicizing of some of their papers on, again, apples, pumpkins, and gourds, all very seasonal crops. For communicators and marketers like myself, we love it when our scientists put out things in conjunction with popular seasons. It really makes it makes it fun. Makes our jobs easy.
Yeah, we should start getting work on like the Christmas tree genome. Or
That's
something.
right, yeah.
Start working on conifers.
You can,
we have like a month, guys.
Conifer, spruce, just
Yeah.
come up with some more genomes for us to put out there.
Oh God, Jesus. Okay, I'll keep thinking about it.
Yeah, he's part of that
Mistletoe,
too.
we gotta do that. That's an easy one.
That's not easy.
Oh, fair enough.
Well, we'll give you all time to get on the same page. And,
Yeah, we'll collaborate.
in the meantime, thank you all for visiting with us. We're going to take a short break, and we'll be back at 12 o'clock we'll visit with members of our postgraduate society here at BTI. Okay, thank you all. You're dismissed.
Yeah.
Go have lunch.